Well, hello, my name is Stephen Ferguson, and I am a scientist in the National Toxicology Program at the National Institute of Environmental Health Sciences. And today I'm going to speak to you about some of our efforts in development of new approach methods for toxicology research. And the title of my presentation is The Power of Resolution, Context, and Physiological Relevance to Model Hepatic Responses to Xenobiotic Exposures. And as a federal employee, I must remind everyone that I have no financial relationships to disclose, and the statements and opinions and conclusions that I make are my own and do not necessarily reflect those of the NIEHS, NIH, or the U.S. government. So many of you may be familiar with the Tox 21 program. I'm part of that program, and the program's been going on since 2005 to work towards using innovative strategies in toxicology research to help us better understand molecular pathways that are being interacted with environmental and other reference chemicals. And that program's been going on for some time now. And while it's had great success in many ways, one of the biggest challenges that has been raised with the program is the lack of xenobiotic metabolism. We know in the field of toxicology that metabolism is an essential process for many compounds in terms of detoxification and metabolic activation. And so the vast majority of the new approach methods that have been used in phase 1 and 2 of Tox 21 and related areas have been devoid or very limited in that and presumed to be limited in that. They also, the pathways that are covered have been limited to what was commercially available or what had been developed in academic labs, focusing on individual pathways rather than necessarily integrating those pathways in a more physiological context towards progressions of toxicological response that might manifest in apical outcomes in toxicology. And then also most of the assays were addition-only assays with minimal ability to look at longitudinal or through time studies to understand the progression of responses. And so one of the things when I came into the program was to try to say, well, where are we in terms of xenobiotic metabolism? And I come from an area that's focused on using liver cells derived from human liver to predict drug metabolism and drug-drug interaction potential. And when you look at the metabolic activity in suspensions of primary human hepatocytes derived from the liver, which are shown in the very tall bars on the far left of the bar graph, you can see the metabolic activities, in this case specific activities for clinically relevant substrates, phenacetin, bupropion, and midazolam, far exceed those that we saw in the legacy Tox 21 assays down in the low regions. And even though you might use more physiologically relevant cell types such as primary hepatocytes or hepRG cells, in culture over 48 to 72 hours, they fall well short of this bar. And so our goal in the program was to attempt to confer xenobiotic metabolism. Now, the challenge is Tox 21 is largely used in mortal or cancer cell lines, such as HepG2 cells, that do have some limited and skewed capacity to metabolize things. But when you look at the structure of the human liver, you see that it's a complex structure, and the complexities and the dynamics that are going on in these tissues are far exceed the complexity and modeling of cells that are cancer cells that are sticking on plastic and heavily proliferating, because that proliferation can impact their ability to model metabolic competence. And so the most successful models have been those that are derived directly from human liver, as you see on the upper left, and the cells in suspension form directly isolated, or those that have been cultured to try to model more chronic responses, have been successfully used to predict quantitatively metabolic clearance, metabolic or uptake clearance in the liver, as well as liver enzyme induction and inhibition and transport, including biliary transport, to be able to integrate data from these in vitro systems into pharmacokinetics models that enable us to estimate and in some cases predict the potential for drug-drug interactions and drug clearance rates and mechanisms. And in many cases, those data are used for decision making to decide whether clinical trials are necessary, or simply the in vitro data can be used for drug labeling. So these become an opportunity to leverage this kind of approach in the Tox 21 program. But it's important to understand that when the primary hepatocytes are ripped out of the native context of the human liver, they begin to rapidly de-differentiate, and the metabolic competence that's so strong in these suspension cells only has a half-life of 1.5 to 3 hours for major clearance enzymes if the cells are simply maintained in a suspension at 37 degrees. But if the cells are cultured, they actually can retain the metabolic competence that they expressed in vivo for roughly 24 hours. And you see this little window of time where the half-life is actually extended for these enzymes about tenfold. And so now you have a half-life of about 20 to 30 hours. And that's an opportunity, but it's still, the system begins to de-differentiate as it adapts to this new cell culture environment. And it's still limiting for us to be able to model metabolic activity when we're only achieving about 10% of the metabolic competence that we had in the liver. And so the Tox21 program is really bringing online these models as well as other models that get closer to the bar of metabolic competence and integrating improved cellular differentiation and functionality models, ones that confer metabolic competence for both detoxification and metabolic activation. They have superior longevity. So, for example, primary rat hepatocytes in 2D culture might last for 3 to 5 days in a standard sandwich culture configuration, whereas in 3D models and flow models, you've seen cells that can last for weeks on end. And in our lab, we've actually done it even longer. I'll show you some examples. And then ultimately, for our program, we're trying to build relationships between our rich legacy of in vivo data to understand what the cross-species parallelogram looks like when we try to relate human in vitro to rodent in vitro to rodent in vivo, and then build that context out with pharmaceuticals and other compounds that are consumed by humans out to understand the clinical or the human environmental exposure, that we can ultimately build those relationships that build confidence in the new approach methods. But it's important that any of these methods be able to model the biological response. That includes, for the liver, a metabolic activation to better mimic the exposures that are produced internally in vivo. So in addition to that, we want to bring online multi-dimensional assay platforms. So you have a model systems that's modeling the complexities of all this these processes, but you want to be able to bring online various technologies to let you image what's happening, to let you understand the genomics or the molecular level changes that are happening in an adaptive way, as well as metabolomics, where things are actually changing, and we can begin to relate the changes that are happening in these various molecular pathways to broader context of understanding cause and effect relationships. And it's critical that in these approaches we leverage the power of resolution that's offered with higher throughput systems so that we can monitor the time and concentration progressions that happen in these systems. Ultimately, in order for any of these new approach methods to be successful, they have to have quantitative translation to humans, and so building bridges to internal exposure concentrations, C steady state, C max, with in vitro potencies, and having a common calibrated method for scaling that, as well as extrapolating that out to the external dose is key. And then ultimately, as we gain confidence that a model system is able to predict a single person's response, we then expand that out to populations and disease states, as well as other culture models like kidney and intestine are two of the ones that we're focused on in the near future. So for our program, we've actually focused predominantly on primary human hepatocytes and this cell line called HepaRG cells. So it's important that when you put these cells in these more complex systems that they are able to differentiate and recover some of the in vivo-like characteristics that they had. And these cells are particularly interesting because they derive from a patient who had hepatocellular carcinoma, although they're not clonal, and also had hepatitis C. The cells are negative for hepatitis C. But what happens with these cells when they're put in culture for about 4 weeks under a differentiation protocol, somewhat like stem cell differentiation, they will actually right along the stem cell differentiation tree and produce co-cultures of hepatocyte-like cells and cholangiocyte-like cells that you can see in the upper middle figure here, and that's showing you staining with CDFDA staining. So this is a particular molecule that highlights the biocanalicular domains that form when these hepatocytes form nice epithelial layers, and they'll begin to polarize and form bile ducts, essentially bile pathways. These are biocanaliculi, but they're starting to increase levels of bile salts and bile acids, and they form these bile pockets in response to that. And you can actually stain for this as evidence that the cells are polarizing and beginning to model more mature liver function. And you see those analogous structures are also produced in human hepatocytes. But the cells have, these HepRG cells have effective transporter expression and function, as you saw in this figure. Their drug metabolism activities mimic the ranges that you see across hundreds of donor preparations of human hepatocytes. And most importantly, they're really the only cell line that's been published to date that are able to support CAR translocation. So CAR is the constitutive androgen receptor, and it's translocation of CAR from the cytosol to the nucleus in response to phenobarbital is a hallmark of hepatocyte functionality. And so these data show that it works and that the induction fold data you see in the bottom panel are analogous to what you see in human liver. Now, what we like most about the cell type is it's our first sort of tier, and it has year-over-year availability in the same donor background. So many of the challenges with primary human hepatocytes are there's a finite number of cells, and when a donor preparation's supply of cells is reached its end, you really can't do any more experiments in that background. So we like the idea of these HepRG cells. For that, they also have reduced variability, and they're much easier to work with than primary hepatocytes. They're somewhat less expensive, and they also have a unique ability to adapt and transdifferentiate. And so if you cause some type of injury or damage, they're more likely as stem cell type cells to be able to repair those areas. And you can see the schematic here that was published in Nature Reviews that basically just shows you the highway of stem cell differentiation that these HepRG are riding along, going from progenitor cells down to hepatoblasts and hepatocytes and cholangiocytes. That's what's thought to be the case, although there's still much work to be done in this area. Now, in our lab, when we started figuring out which systems we were going to look at, it was important for us to have a screening model, something that we could run 3D 4-well plate assays to get the throughput we needed to interrogate time and concentration effects across sets of chemicals, roughly 10 to 100 chemical range numbers of chemicals. And we tried different systems, and there are lots of systems available. The system we ended up settling on in the initial publications were these Corning ultra-low affinity microplates you see in the middle of the screen. And the very first plate that we ever tried produced very nice spheroids, and this is actually the image from that very first plate. And it worked perfectly. Every well produced a sphere. We were able to feed the media and chains of cells with a ViaFlow 3D4 liquid handler. And we extended that work out into 3D 4-well plates. What's really nice about the system is that it uses far fewer hepatocytes. And so we see primary hepatocytes and HepRG as a gift to the research community to be able to study liver function. And it's basically a tenfold more efficient use of cells if you can get the endpoints that you're looking for. And so instead of one vial leading to one 96-well plate, now you can get 50 to 100 plates in 3D using these configurations, and in the 3D you can get roughly 12 to 25 plates, as you see here. But this is a work that was led in the lab by Srinivasa Ramagari, a very talented postdoctoral fellow who's now actually been promoted to research fellow within our program, and it made the cover of Toxai. You can see across the plate nice consistency across the plate. And in the lower right-hand panel, you see the size metrics just confirming the micron sizes of each of the spheres across the plates. And so we're confident that we're able to reproduce this consistently. When we did immunohistochemistry staining and collaboration with Darlene Dixon, a molecular pathologist in our branch. We were able to see H&E staining that was consistent with good quality hepatocytes that are forming nice cell-cell interactions. Glycogen staining was confirmed by PAS staining, so that the spheres are able to store glycogen. Poly-CEA and MRP2 staining confirmed what we said earlier about the biocanalicular domains we saw in 2D are actually also produced in 3D. And interestingly, we saw over time that the proportion of CK19 positive cells, which are shown in the upper right-hand corner, actually shifted the numbers of hepatocytes, or the proportions of hepatocytes and cholangiocytes. So CK19 is a marker for cholangiocytes in liver. And you can see here they tend to form a sort of exoskeleton around the sphere. And we saw this repeatedly, and other labs have seen this as well. Magnuson, Engelman, Sunbird's group showed this very nicely in primary human hepatocytes that have non-parenchymal cells available in the lower panel that you see here. And so it's encouraging that this is matching the types of responses and the types of morphology that we see with primary hepatocytes. In the early days, the spheroids, as they're maturing, the CK19 positive staining tends to be a little bit more distributed through the sphere, but over time it tends to be highly localized on the periphery, as you see here. So the HepRG are certainly going through some adaptive response. So this image shows you MRP2 staining, immunohistochemistry staining, confocal imaging, and you can see slices through the spheres, and this basically is confirming not only do these HepRG spheroids form biocanalicular, but the networks that are formed are much more extensive than what we saw in traditional 2D cultures. So it's very encouraging. We're seeing this. This is definitely a good sign. We don't see any evidence of necrotic centers forming, and you can see the Z sections as it's scanning through, that you'll get these much more extensive networks to be forming. And so this is evidence that the efflux transporter, the biliary efflux transporter, MRP2, is localized in the proper domains in these spheres. Moreover, when we look at cholelysylfluorescein, which is a substrate for MRP2 and the bile salt export pump, we can see live cell imaging that shows that dye is matching the types of internal networks that we saw in the MRP2 staining. And you can see also the very high levels of CYP3A4 immunoreactive protein. So we started to figure to ask the question, well, how do these spheres match up with other models in terms of the metabolic competence spectrum that I mentioned earlier, and trying to achieve things in the Tox 21 program. And so this is sort of this is your life of metabolism as I've gone to the different model systems and cell types. And you can see here the median suspension hepatocytes on the far left of this plot, sort of representing the benchmark that's approximating human liver. Again, Tox 21 cell lines very low. Interestingly, these HepRG cells, even though they're great cells, if you culture them in proliferating mode, they actually don't have metabolic competence that is even close to the goal that we're trying to achieve in suspensions of hepatocytes. And actually, for CYP1A's case, CYP1A2, the levels are relatively comparable to HepG2 cells. But when you culture those cells in a more differentiated state, in 2D differentiated state, you can actually see the activities increase about tenfold. And then when you culture them in 3D spheroids, you actually get very close to the benchmark. And these two conditions are no DMSO on the far right, or 0.3% DMSO in the next most in the PIT on the left side of that box. So what these data are telling us is these little small spheres of HepRG cells are able to achieve sufficient levels of specific activity that rivals the activities observed in primary human hepatocytes. Now, my first concern was, well, if we've got very high activities, maybe we've just induced these things by having them in 3D. And I was very happy to see that not only were we able to achieve high basal levels, but we were also able to see further inducibility of CYP1A2, 2B6, and CYP3A4 mRNA and enzymatic activity. In this case, we're showing the mRNA with classical activators of the aryl hydrocarbon receptor, the constitutive antigen receptor, and the pregnane X receptor. And so these are, again, hallmarks of hepatocyte functionality, and what they're confirming to us is that not only is the system basal metabolism very robust, but it's also analogous to the liver, able to functionally respond to classical receptor pathways for the liver that upregulate expression and enzymatic activity of these pathways. And so this is what we want to model in terms of liver. So we're very excited about these models and their ability to be able to, as we say, have our cake and eat it too on the metabolism and the receptor signaling pathways. But there are lots of challenges, and we'll talk about those as we go along. One of the things in the paper that we published a couple of years ago is basically showing aflatoxin B1, which is metabolically activated. And you can see here that compared to 2D HepRG, which is showing up in the right, the least potent curve across this group, as we switch to 3D spheroids, we see a left shift in the potency. They become more sensitive, which is consistent with what we'd expect. The IC50 shifts from 46 micromolar down to about 8 micromolar. And then by doing repeated exposure, we shift that even further to the right, so doing more damage. And to get a sense of whether this was related to metabolism, we use 1-aminobenzotriazole on the lower right-hand panel, and you can see we were able to markedly attenuate the cytotoxicity that was observed with aflatoxin by doing that. So even just a simple cytotox evaluation when you have this kind of metabolism can be a very useful tool. And we actually showed that in the paper that included looking at drug analog case studies where the one drug analog, in this case trovafloxacin or troglitazone, was withdrawn from the market due to liver injury. And what we did was compare those liver injury compounds with drug analogs that weren't as often associated or weren't associated at all with liver injury. And what you see is just using a simple cytotox assay and repeated exposure with the 3D spheroids, we were able to pick up these molecules and might have done a better job with these kinds of data and flagging this for additional focus before they were released in the market. So we think this is a valuable tool to begin to apply in various drug and environmental chemical estimations of liver injury. Not only do we have the model for human primary hepatocytes and HepRG, but we've also been working with rat hepatocytes, as I said, for the parallelogram approach, and Srini's been doing a great job in leading this effort. You can see on the lower left-hand corner, we get similar types of staining to what we saw with the HepRG spheres. So you get this pan-cytokeratin sort of exoskeleton on the outside of the sphere. The metabolic activities, as we might expect, are very strong for the first 24 hours. They drop as the spheroids adapt and recover from this process, and then out to 14 days, they're perfectly happy. We've gone out to 28 days after this, and we do see a slight decrease. It's about 50% decrease in the activities from those levels, but we're still working on things like cell culture media composition to see if there are things that we're doing in 3D spheroids that may be altering that. But we're very excited that for this 14-day window, the model would be quite useful for us. Srini also confirmed that the model is metabolically inducible. So this is treatment with phenobarbital, and you can see CYP2B1 mRNA is quite nicely inducible, again, consistent with the HepRGs and important for us in the toxicology field. And then liver injury compound like diclofenac is able to show a loss of viability as you get to higher exposure levels. And so we're in the process of putting this system through its paces with a battery of chemicals and also with high throughput transcriptomics. So the majority of the presentation today, I wanted to talk about our efforts not only with these model systems to try to tackle modeling the liver in some sort of basic way that's useful, but also to begin to explore what are the features that are distinguishing three-dimensional cultures two-dimensional cultures and proliferating cultures from one another to be able to better understand when to apply the various models for different questions. And so to do that, what we've done is chosen a set of 24 compounds as our reference set of chemicals to begin looking at this. And the majority of these chemicals are shown in purple are liver injury compounds, or compounds that have been associated with metabolically activated liver injury. The ones shown in burgundy are hepatic receptor activators, and they're classical inducers or activators of the aryl hydrocarbon receptor, pregnane X receptor, constitutive androsten receptor, farnesoid X receptor FXR, and the peroxisome proliferator activator receptor alpha, PPAR alpha. And so those are all shown in burgundy. We also, analogous to what I showed earlier, we have the 4 drug analogs, rosiglitazone versus troglitazone, and levofloxacin versus trovafloxacin. And then finally, a set of compounds that have generally been shown to almost never be associated with liver injury, including aspirin and caffeine and sucrose. So in terms of these chemicals, we ran a series of assays with this study, cell morphology using either an IncuSight Zoom or an Image Express confocal. Molecular devices, using cytotoxicity with LDH leakage in each of the systems every time we change culture media, and then high throughput transcriptomics using a novel platform that we have worked with. And I'll describe more about that in a moment. We also collected supernatants from the study for metabolomic studies, and some of those studies are also being published, but I don't have time to talk about those today. So I want to go into this high-throughput transcriptomics a bit. So one of the challenges in our program is if you try to do whole transcriptome or RNA-seq analysis on chemical screening, it would be an incredibly resource-intensive exercise. And so the idea was to try to focus transcriptomics and genomics research for toxicology applications. And so, led by Rick Paulus and Scott Auerbach and Shirsal Deepak Mav, we've developed a set of genes called the S1500 plus gene set that is selected for capturing diversity. It's attempting to find sentinel genes that sort of maximize our coverage of pathways. And in this case, it started out as roughly 1,500 genes. It then got expanded through nominations into about close to 3,000 transcripts, or 2,977. And then an ultimate goal was to see if you could then take these data and be able to extrapolate it out to what the whole transcriptome would be. And so this is published in PLOS One. You can take a look at that. The highlight would be that we tried to cover all the biological response pathways that were possible in as efficient method as possible, and using the NCATS bioplanet that's recently been released to the public, and you can see that link below, you are able to cover with 4 or more transcripts 77% of the genes in the human pathways annotations that go biological process. But you at least have 1 or more genes in 96% of currently annotated human pathways. So this is a very very focused but efficient way to cover biological space. And the platform is currently offered commercially by a company called BioSpider, and they've labeled the platform TempoSeq. It is a modified RASL-seq, and the set of 2,977 transcripts are interrogated in our approach. And what's really nice about the platform is you can do direct lysis and assay. You don't have to do any RNA isolation, which is a massive hurdle when you're talking about these kinds of studies. And so we seed between 1 and 25,000 cells per well, depending on the assay system. We add lysis buffer to a total of 20 microliters, and then we run the TempoSeq assay. And in our shop, the TempoSeq assays, we don't run it and currently. BioSpider is doing that under contracts with us. But the assay works, and it's been published. You can see Joe Yeakley's paper from 2017 in PLOS One here. The assay works by targeting 50-mer oligonucleotides that are spaced by a bridge that is ligated. That's the sort of razzle, the ligation part of this. And then essentially removing excess oligos and leveraging the detector oligo ligation that happens, you're able to, with barcoding on these oligos, load all of the samples into a few lanes of a next-gen sequencer, and you're able to get map sequences, map read counts for the targeted 50-mer region that you're looking for the 2,977 transcripts that we look at. And you can dig into more of the details there, but suffice it to say, instead of sequencing everything, you're sequencing very targeted and efficient set of alleles, set of regions of these. And so the idea is to understand the relative expression of those map read counts relative to one another. And you have to do that on a high-seq. And currently that's done at Biospider for us, and then they send us FASTQ files. We take those FASTQ files, and we've developed a QC pipeline that looks at sequencing depth, alignment rates, the per base sequence quality, pass-fail flags. We do account for cytotoxicity and looking at the data and flagging it. We're looking at for the counts per million, and we do normalize the data from each well to a common counts per million target, as the efficiencies from well to well can vary. We floor the data that I'll be showing you today at a recount of 5, and we're playing with different floors, but right now we're at somewhere between 5 and 10. We log 2 transform the data, and then our partners at Siome basically finish the QC pipeline by giving us a series of output files that are ready to import to BMD Express, Partec, Flow, and Ingenuity pathway analysis. And we get out after the QC pipeline these map read counts. And here's an example of the QC flags that Siome's providing for us, and you can see the concentrations that we added on these plates were actually high on the left-hand side starting in column 3, and they went to lower concentrations as you got to the right. And so you can get a sense of the types of data we're getting, and certainly when you have cytotoxicity, which is happening on the left-hand side of the plate, you get a lot more flags, as you might expect, as activities and gene expression is lowered. So now I want to go into this study in more detail and kind of some of the things we saw when we leveraged this high-throughput transcriptomics platform. And as a person who's been working in the liver enzyme induction world for the majority of my career, the first thing I wanted to do with these data was to look at reference sentinel genes, specific transcripts that I'm very comfortable with liver models, and understand do they show the same types of responses that you see with a conventional TACMAN assay. And what you see here on the upper left is the expression of CYP1A1 that was in the S1500 array. And you can see in the black curve is the HEPA-RG cells in proliferated mode, and they had a nice response to the AHR activator omeprazole. In 2D cultures, you can see omeprazole also produced an induction of CYP1A1, but the basal levels for CYP1A1 were much lower in 2D relative to proliferated. Now this actually makes perfect sense, since we know that CYP1A1 is overexpressed in developing liver. And so this actually makes perfect sense. And then looking at a different facet of omeprazole response, we know that omeprazole is metabolically activated by CYP3A4 to a sulfone metabolite, shown on the lower left in red in a cartoon. And that metabolite is thought to be an agonist for the pregnant X receptor. And so that metabolically activated response would then lead to a PXR activation and induction of CYP3A4. So on the lower right-hand corner, if you look at CYP3A4 induction, comparing the proliferated versus the two-dimensional differentiated HEPA-RG, you see that the 2D differentiated had far superior metabolic activity, induction of CYP3A4 mRNA by activation through presumed to be through activation of the pregnant X receptor, compared to the proliferated cultures that had far lower constitutive levels of CYP3A4 mRNA, as well as smaller inducibility. And we saw that across the board when you look at the spectrum of pathways, the sort of reference pathways for AHR, CAR, PXR, PPAR, and FXR, you can see genes that are sentinel to those pathways generally produced a larger dynamic range in the 2D differentiated cultures, and interestingly, also somewhat less shifted potency. So the proliferating cells tended to be a little bit more sensitive, and this could be related to in vitro disposition and other factors that we are as yet unclear on. But what we do know is that the 2D differentiated models are working properly, and these data confirm that these pathways are working analogous to the types of studies we would run for liver enzyme induction predictions for regulatory studies. So the 3D cultures, the first batch that we ran actually failed, and we're still not quite sure. We haven't been able to replicate the failure. But since that time, we've done lots more work with the 3D cultures, and you can see here very nice inducibility of CYP3A4 and CYP2B6 in response to rifampicin and phenobarbital in the 3D cultures, looking at tempo-seq data, and then doing ingenuity pathway sort of untargeted analysis. CAR and PXR pathways essentially populate at the very top in response to these activators. And so this is very encouraging that we're seeing the things we're supposed to see in an enriched way. Now, our program has moved towards not only using these types of unsupervised approaches and these individual genes that are really reference genes or sentinel genes for a pathway, but starting to look at more of a global pathway analysis. And to do that, Scott Auerbach in our program, in collaboration with Scient, has generated the next iteration of BMD Express, and this is BMD Express 2.0. This package is very effective and efficient in taking various types of data and doing dose response modeling. And what we've done after a workshop that we had to try to understand the best approaches for analyzing these data is that we've actually leveraged the power of concentration response modeling to filter the data. So we first run a Williams trend test on the data to see if we have a dose response curve, and then we filter that based on a fold cutoff, usually 2-fold or 1.5-fold. And then we do benchmark concentration analysis, and that's essentially, for those of you who aren't familiar with it, it's looking at the point of departure. So you have a curve, and you have the vehicle controls variation, and then you have a dose response trend. And if it has a dose response trend and there's a statistically significant point of departure that you define in the software, then we call that a benchmark concentration curve, or BMC. If you're doing in vivo, it would be a benchmark dose, so BMD, which is where the software was originally designed. And so in our case, we're leveraging this software to basically identify dose response relationships in the data and then summarize that in what is referred to as accumulation plots. So you can consider this somewhat like a concentration resolved histogram. And so on the upper left, you see the response for one gene. That's a classical way of looking at a dose response relationship. The point of departure for that curve would then be one of the circles on the accumulation plot shown on the lower right hand side. And what we see when we start to look at this dose response histogram is that genes that are associated with the protein X receptor, those they're painted in red in this particular plot that I did manually before we had the ability to paint pathways, are actually all showing up on the left-hand side of the dose response, and at concentrations that are below the maximum therapeutic concentrations that are known in humans under the classical exposure levels. Meanwhile, pathways related to stress, and ER stress, oxidative stress, apoptosis that have historically been associated with rifampicin exposures as well are all showing up on the right-hand side of the curve. So you're starting to see this ability to resolve sort of off-target or pharmacological pathways from things that are happening at higher concentrations that might be stress, death, very deleterious pathways. And so we believe that the concentration response modeling and the filtering of the data enables us to have this power of resolution to start distinguishing out the sort of intended and unintended effects of chemicals. And we've leveraged this across the full set of 24 compounds, and this is published in our June paper in 2019 in Toxai, where we took this set of 24 compounds and essentially generated these benchmark concentration analyses and accumulation plots for each of the chemicals in this set, and it was run 3 independent times. This is one of the runs. And what you see is compounds like aflatoxin, chlorpromazine, troglitazone, acetaminophen, trovafloxacin, ritonavir, benzoylpyrene, all the liver injury compounds are just blowing up gene expression. You see these sort of hockey stick type appearances, and this is in line with the general thinking around genomics and cellular stress and death response. What's nice about this system is it has the resolution to not only tell you if something could be a liver injury compound, but we're able to use the data to define a threshold for liver injury estimation. And that threshold was 3 standard deviations above the mean of the presumed rarely associated or negative compounds that we've published in the paper. And so what you end up seeing is the point at which the accumulation plot crosses the threshold is an estimation of the relative potency of that molecule to cause liver injury in humans. And when you start to look at where those concentrations are in comparison with where liver injury is observed, you can calculate what I termed a CILI index, or a chemical-induced liver injury index, that's 10 times Cmax divided by the threshold concentration, which is called BMC 105 in this case, because the 105th gene that was changed was deemed the statistically driven threshold for this data set. Now, that'll be a moving average as we add more chemicals to this approach, but it's an untargeted way to rapidly screen and understand if a chemical has liver injury potential and at what concentrations you might start to be more concerned about that for follow-up studies. So we're really excited about that. In addition to the potency estimations, you can also begin to integrate other data types. So in this case, we see morphology data that was collected in the study, and all of these data have been published, and they're all publicly available, and you can go to the publication and see those links. But we host it here in a database called SEBS, as well as the traditional genomics databases as well. But in this case, the images, you can look at all the images yourself. This is an image on the far left showing the proliferated versus the 2D HepaRG in culture. We use an InkySight zoom to get these images for every single culture well at multiple times throughout the exposure period. And you can see here highlighted are hepatocytes and cholangiocytes. In the 2D, you can't really distinguish those cell types in the proliferated mode very effectively. But for 96 hours, you'll see that the 2D cultures looked comparable, whereas the proliferated cultures, you can clearly see that they proliferated during the exposure period and become virtually confluent after 96 hours. Now, when we treated with chemicals, we saw many cases where that wasn't affected at all, and then as you got to higher concentrations, you would often see cells die and you would lose all the cells on the culture. What was interesting, though, is when you start to look at some of the transition zones, like 1.5 micromolar aflatoxin, we actually saw areas where you saw attenuation of proliferation, because you see less cells here. So either the cells were dying or cells weren't growing as fast, which is consistent with what we know about the mechanism of aflatoxin B1. But moreover, in the 2D cultures, you can actually see evidence of scarring, where cells have died and the other cells and these are presumably cholangiocytes that are trying to fill in the gaps and trying to repair and do damage repair. And this actually shows up in the pathway analysis. It also shows up in the LDH leakage data. So you can see the sort of threshold that we're looking at here, 1.5 micromolar, was right at the edge where you started to have clear cytotoxicity. And again, all these curves are available as well for your evidence, for your further look. But the take home is that these models, as you start to integrate cell viability, cell morphology, and what I'm going to show you next, transcriptomics, you can begin to get a more contextualized understanding of what's happening in response to the chemical exposure that not only informs you about potencies, that can also inform us about what's going on. And so in this case, this is pathway analysis using Ingenuity pathway analysis, as well as benchmark concentration modeling on the upper right-hand corner, that reveals the types of pathways we would expect to see with aflatoxin: apoptosis, p53 signaling, DNA damage, cell cycle. And comparing 2D versus proliferated, we saw the relative trends we would have expected to see with those molecules, both in terms of aflatoxin as well as another genotoxic agent, benzoalpha-pyrene, which is also metabolically activated. And the sort of take home from all of this is we're starting to build relationships now between genomic or transcriptomic pathways and phenotypic changes that are happening in the cell cultures to build confidence about those relationships and our ability to interpret the genomic data. Now, one of the really cool compounds that we took a look at, and one of the most difficult to model in vitro with less metabolically competent systems, is cyclophosphamide. And this just captures a high-level summary of that. But when we cultured the cells in proliferated mode, we saw virtually nothing, which is consistent with the fact that cyclophosphamide requires CYP2B6 activation to form the ultimate cytotoxic phosphoride, mustard, and acrolein that are a bit more stressful. In 2D cultures, in the publication that we showed originally, we got decent modeling, and you can see we have some response here that's starting to show pathways that actually made sense. But when we were able to generate the 3D data relatively recently, and these data are still unpublished, we saw a massive increase in the amount of transcripts that were observed. And when we looked at the pathways, they make sense. Lipid hydroxylation, P450 metabolism, and as you go to higher concentrations, you get into cell cycle, oxidative stress, DNA damage, hypoxia, which is actually observed in elderly patients who are exposed to cyclophosphamide in many cases and start to have issues in maintaining the oxygen levels in their blood. So, we're starting to leverage this information further. We need more work to understand it better, but the 3D model seems to really be doing some very special things in a metabolically dependent state that we want to test further, add inhibitors, and do follow-up. So in the publication in Toxine 2019, we actually also looked at the trovafloxacin versus levofloxacin comparison, and you can see readily the massive distinction between levofloxacin, even though it had higher therapeutic exposure levels, versus trovafloxacin. Both of these molecules were designed to be used in therapeutic treatment of pneumonia. And so you would imagine most of the effects, you would hope to see very little effects on human liver. And in fact, we saw a pretty massive response with trovafloxacin that would have given us pause to move that molecule forward. And similarly, if you look at the pathway level instead of the gene level, you get lots of pathways related to cell killing, apoptosis, DNA damage. You really see none of that with that levofloxacin using the same filters. So all in all, very promising, and the fact that we can begin to distinguish clinically relevant drug analogs that have been used and have different outcomes in humans, it gives us more confidence that we're headed down the right path. Now, another case study that I want to talk about is troglitazone versus rosiglitazone. So the transcriptomic pathway analysis was able to readily differentiate these molecules, analogous to the trovafloxacin versus levofloxacin analysis that we did earlier. And you see the cell stress and damage pathways painted here, where rosiglitazone is far less potent than troglitazone, which is shown in the 3 independent runs on the left set of curves that are sort of sloping. Interestingly, this is one of the few molecules we had in the set where we actually had the ability to start looking at therapeutic target interactions. And when we did that for PPAR gamma, in the case of these glitazone molecules, we saw a very puzzling relative potency. So troglitazone, which would be on the left-hand set of curves, that showed PPAR gamma potencies that were far more potent than pathways that were identified in response to rosiglitazone. Now, that's completely reversed from what we know in PPAR gamma reporter gene assays, where troglitazone is in fact about 550 nanomolar potency versus 18 nanomolar with rosiglitazone. And this was quite puzzling at first for me. But one of the things that we can do in BMD Express is start to look under these data and start to say, well, what genes are making up these pathways and how are they responding individually? And so you can do these curve overlays in the package. And it was readily obvious that if you look at highly specific selective compounds, genes that are in the PPAR gamma pathway, like adiponectin and fatty acid binding protein 4, the relative potencies between rosiglitazone and troglitazone match perfectly. What's happened is that, likely due to the lack of ability to resolve cell stress and damage pathways versus more close focused therapeutic targets, you're actually getting the median BMD, which is plotted on that original plot, is being pulled by the transcripts that are basically producing responses at the very, very highest concentrations. And so that median is being shifted due to what is likely a confounding effect on cellular stress pathways. It's possible that also currently annotated pathways aren't universally applicable to adipose versus liver, and we need more work to try to understand that. But the general presumption in the field is if it's a pathway, it's always a pathway across all. So the pathways that are currently annotated may not be universally applicable to all cell types and cell contexts. Another example that we've just generated, and it's really exciting data that we need to repeat multiple times to make sure, but one of the big failures in the 2D versus proliferated study that's been published was valproic acid. We presumed that because we did not achieve levels that were 10 times Cmax at our highest concentration, that we likely just didn't go high enough in exposure, and that may be true. But when we did the 3D HepRG with the valproic acid and looked at those same concentration ranges, it was a massive increase in pathways. This molecule is metabolically activated. And what we saw was low concentration effects on fatty acid biosynthesis and mitochondrial fatty acid beta oxidation that are consistent with what we know are effects with valproic acid on lipid levels and lipid homeostasis. We actually saw an interesting effect on the GABA receptor pathway, which normally you don't think about in liver, but it looks like we were able to clearly identify that, and we need to do this multiple iterations, but it's possible that even though we're in liver, we may be able to pick up effects that would be predictive of therapeutic effects in other tissues. And then as you get to higher concentrations, we know that idiosyncratic liver injury and hyperammonemia are observed, like that's been associated with mitochondrial toxicity. And as you get to the very highest levels, you can see urea cycle, cell cycle mitotic effects that are popping up. And so this is very encouraging. We're really excited about it, and we're hopeful that this is going to be much more powerful as we go to the full set of 24 compounds in the next couple of months. Another example, Rifampicin comparing 2D versus 3D. We see that both models are able to model CAR and PXR signaling. You can see, and we also get higher exposure level PPAR signaling in 2D. You see the same pathways in 3D that are lighting up. So the 2D model's working. But what we think may be happening, and you can see it manifest in additional pathways are lighting up in 3D, that they may be modeling a more comprehensive set of biological responses. And we're not sure. It's possible that this is more related to in vitro disposition differences between 3D and 2D. We have projects that are starting to dig into that question and understand it. But when we took the data, and Srini took the IPA analysis and compared the benzoylpyrene pathways, P53, AHR signaling, hepatic fibrosis, lots of different pathways, and you look at 3 micromolar exposure in 3D versus 2D versus proliferated in the 3 columns, and what you see is that you have a clear enrichment of genes within a pathway in 3D relative to the 2D and proliferated mode. So it's quite possible that 3D are just better mimicking the pathways that have been historically annotated in vivo and from other data sources to give a more comprehensive sort of modeling and capturing of the dynamics that are going on in the system. Now, he also is happy to point out that the pancreatic adenocarcinoma is identified as one of the pathways here, and that has been associated with benzoylpyrene in humans. So I wanted to point that out. Now, while we have a lot of opportunities with the 3D, I've tried to capture some of those long-term culture ability to model more complex metabolic activated responses, more efficient use of cells. There are lots of challenges. We've had recent issues with plate manufacturing that we're not able to always get the plates that are working for us. Changing the culture media can be a challenge if you don't have the right pipette systems in place. We're still trying to understand the unique properties of 3D. So culture media has been evolved from 2D cultures that generally are not have not been optimized and tuned for 3D cultures, and we're starting to see some differences that may suggest that there are some needs to go and to look deeper, and we're starting to look at that now to see if there's some things. Some of the factors include DMSO, hydrocortisone, insulin, glucose. And then probably the biggest issue with these particular models is you're dealing with a very small number of cells per well. So trying to scale the biomass of that small amount of cells per the media volume into quantitative translation to humans needs calibration. And so we're going to need work to maybe titrate the numbers of spheres and things like that to better mimic, or to come up with informatic ways to adjust the data depending on the data stream that we're able to achieve. Now, one of the things, Caitlin Lavrich is a new postdoc in the group, and one of the things she did was start to culture these things out for very long periods of time. And she saw a very disturbing but maybe exciting opportunity with HepRG cells, and that was when you culture them with no DMSO, 0%. For 56 days, you actually saw they more than doubled in size and density, and I'll show you the plot. And she used 0.3% DMSO, and she actually saw that effect went away. So you can see the density shift is much more attenuated. Now, these HepRG cells were derived from hepatocellular carcinoma, so it's possible that they have some cancer properties that are manifesting in these systems over time. We know they do not grow in nude mice. It's been published. We've heard that they will grow somewhat in soft agar, so that we presume that was more of a stem cell property. But at this time, it may be an opportunity to start to look at molecules that could disrupt this effect and maybe start infer some ability of molecules to interact. We know that DMSO has been associated with AP1 signaling, and so that tumor suppressor type pathway might be operative here. We're definitely going to dig into that more. And Caitlin's been not only looking at that, but looking at the baseline genomics between our systems and also the bioenergetics between the systems to try to understand what is really unique between 3D and 2D systems. And what she's finding in the gene expression is that the 3D are enriched in things we've been talking about, xenobiotic metabolism, bile acid metabolism, but they're also upregulated in oxidative phosphorylation. Meanwhile, 2D cultures seem to be enriched in epithelial mesenchymal transitions, so evidence of back differentiation, hypoxia, inflammatory response, and glycolysis, which is intriguing in that maybe 3D cultures might actually be more aerobic, while 2D cultures are more anaerobic, and more work is needed on that. But preliminary data from SOT suggested that the oxygen consumption rate in spheroids of primary human hepatocytes was far greater in 3D than it was in 2D. So more work to be done there. But if energy is being used different in these systems, culture media may need to be optimized as well. So in summary, I hope I've conveyed to you some evolving approaches that we're using here in the Tox 21 program to improve the physiological relevance and the ability to model xenobiotic metabolism, and to begin to bring online assays that don't just cover one pathway at a time, but cover a broader spectrum of pathways. And we're hopeful that future models are going to allow us to expand this concept out to a broader set of toxicological tools and new approach methods. I hope I've convinced you that high-throughput transcriptomics with this platform can be a very efficient survey of responses and allow you to rapidly gain an understanding of potency and potential for liver injury, and then to begin to look under the hood to understand what may be happening in a manner that leverages the resolving power of concentration response modeling to distinguish the effects of lower dose exposures that may be more pharmacological versus the off-target and higher exposure levels that may be starting to get to cell stress and death, and understanding the context of where you are in that system. And then finally, an upcoming focus on maturation, kinetics, and time response and reversibility is currently going on in the lab. With that, I do want to highlight, I know I highlighted Srini Ramagari and Caitlin Lavrich for their work, Scott Auerbach as well in his pioneering work with the BMD Express 2.0, Rick Paulus in his efforts with Siam and Scott and Rashir Shah and Deepak Mav to get us the the S1500 array, Mike DeVito, who's our branch chief in NTP Labs, and all of our colleagues there who are working diligently on the various model systems to try to develop better methods and understand chemicals that we're being exposed to, as well as Alex Merrick and NIEHS Sipes and their work, Trey Sadler as well, who helped a lot with the transcriptomics. And I always want to mention Ed Lecluse, who taught me everything I know about primary liver cells. And with that, I'd be happy to take any questions.